Hi guys, this is Dr. D back again, and we are going to talk about macromolecules. The macromolecules we are going to talk about today are proteins. Alright, proteins are very large nitrogen containing compounds. They are the largest and the most complex molecules that are found in the cell, and more different type of protein molecules than all other type of molecules combined. I'll explain why that is possible. And they have the following functions. This slide is very simple. I'm reading from the slide. Well, first function, of course, is that they have a structural function, and that is that they make up collagen, which is a connective tissue like the silk in the spider webs, keratin, that's in the hair and the horns of animals, actin and myosin in our muscles. So these are the structural proteins. What about enzymes? We are going to talk about enzymes later. For now, it's important that you understand that enzymes are catalysts. We call enzymes as biocatalysts because these catalysts, they change the speed of the reaction. So they change the rate of reaction. I'm going to write a reaction as Rx. Example, urease is an enzyme that will help us form urea from ammonia and carbon dioxide that's released in the urine. Alright, then there are transport proteins like the carrier proteins. Carrier proteins are found in cell membranes. We will see this in the next couple of chapters. These are proteins in the cell membrane, and these carrier proteins will import certain very specific molecules in and out of the cell. These are carrier proteins. Defense, blood antibodies. The antibodies in our blood are made up of proteins. You study this in Biology 202 in great detail later on, and these antibodies provide immunity to our body. And last but not the least, the proteins, also make up hormones. Hormones, like example, insulin. Hormones, what happens is, have a big influence on the metabolism of the cell. For example, the thyroid hormones. The thyroid hormones will increase metabolism. So those who do not have or do not produce enough thyroid hormones, they have a very low metabolism. Hormones and enzymes are different. Please keep that in mind. Enzymes will change the rate of one reaction. Hormones have a big influence on the overall metabolic activity of the cell or the organ. These are amino acids. Now it's ridiculous for you at this stage to be studying all the different types of amino acids. There are about 20 amino acids. Well, what do you see? All amino acids have a carbon. Right here is the carbon, is a central carbon. The carbon is attached to, you notice, to an NH2. This is the amino group. And also here is the NH2. That's an N, by the way. Here is the hydrogen. Now they also, well, I'll write the hydrogen here. I will write the R group here. So here I'm going to write as the COOH. This is how amino acid looks like. So carbon in the middle. I'm going to write the amino group NH2 here. The acid group COOH here. There is a hydrogen here. And then there's an alkyl group R. Let's look at the R group. So all amino acids, whether it's serine, threonine, methionine, cysteine, asparagine, glutamine, you do not have to know the structure. What is the basic structure of all amino acids? They have a carbon in the middle. They have an acid group, COOH, amino group, NH2. They have a hydrogen. And what makes these amino acids different is the alkyl group. Look, this one has CH2SH. This one has a different alkyl group, CH2, CONH2. You do not have to know these. You have to know the basic structure of an amino acid. That is what I'm going to hold you to. Okay, there are about 20 different amino acids. So imagine a combination of these 20 amino acids makes a protein different, because as you recall, proteins are made up of amino acids. For example, English language, there are 26 different alphabets. So because of these alphabets, we can make so many different new words. Same way, we have 20 amino acids. Combination of these 20 amino acids make up different protein. All right, these amino acids are joined by by removal of water. Okay, that is called the condensation reaction. So here, when you remove a molecule, so this is one amino acid, a second amino acid. Again, you are noticing the R group, which is making this amino acid different from this amino acid. All right, so here there are two amino acids. All I'm doing is removing a molecule of water, OH from here, H from here. If I remove this, now the two amino acids are joined together, and this bond that's formed, the CONH bond that's formed, is called the peptide bond. So basically, proteins are made up of amino acids joined by a peptide bond. How do we get this peptide bond? Simple. We remove a molecule of water. Now, if we want to break down a protein, then all we have to do is add back the water. So here that is hydrolysis. Hydro means water, lysis means breakdown. So if I take a protein, add water here, you are going to get the two amino acids, 1 and 2. Once again, two amino acids can be joined by a condensation reaction, removal of water, and you are going to get a protein. Well, out here, two amino acids joined by a peptide bond. Many amino acids joined by this peptide bond, as you can see, will give you what is called a protein. All right. So a protein structure, to have a protein structure, first of all, you need to understand is that proteins are very highly folded structures. Okay, you don't see this with others, but in a protein, they are highly folded. So we have 4 levels of folding. Remember, we are folding the protein so that the protein has a definitive shape. Okay, and this folding is possible because of the hydrogen bonds. All right, so we have 4 levels of folding. A simple analogy would be, let's say, for example, I'm folding this piece of cloth and I'm folding it correctly. So I do the laundry, I fold this. So I'll fold it in the middle, then I'll fold it once again, and then I'll fold it once again, and then I fold it like this. Just for amusement, I'm folding this piece of cloth into a triangle. Now there are certain steps of folding. Same way, a protein is folded. The first level of folding is the primary structure. Primary structure is nothing but a chain of amino acids joined by peptide bonds. So here you see this stick is nothing but a peptide bond, and these are the different amino acids. I've just put numbers for you for now. This is just to show you this is a combination of different amino acids. And of course, this protein is different from this protein just because of the arrangement or combination of the amino acids. Here it is 1, 7, 3, 5, 19. Here it is 1, 3, 5, 7, and 19. Totally 2 different proteins just because I have a different arrangement of the amino acids. So this is primary structure. Straight chain of amino acids joined by peptide bond. Okay. Now we come to the next level of folding. The next level of folding is that the straight chain now gets folded into coils or into sheets. These coils are called the alpha helix. They are called the alpha helix. Alpha can be written like this, or you can write alpha like this. Or you can also fold it into sheets, beta sheets. You can write beta like this, or you can write beta like this. So this coiling is possible, as you notice, because of these hydrogen bonds. So these hydrogen bonds allow for this coiling or this pleating, and these pleats and these sheets will provide the secondary structure of the protein. That's the second step. Now the third step. The third step is that the coiled and the pleated protein chain is now coiled upon itself again. So I'm further coiling and further folding. So here what happens is that there is further folding. This is the third step. So here you use hydrogen bonds. There are disulfide bonds, that is between S and S, disulfide, two sulfide bonds. And these help to form a tertiary structure. So at the end of this, you are going to get a folded polypeptide. Okay? You are going to get a folded polypeptide. And finally, you have the quaternary structure. So quaternary structure is made up of many or two, I wouldn't say many, I will say 2, 3 or 4 polypeptides chains. These are pleated and folded polypeptide chains, and now they are going to be joined together by some kind of a cofactor. All right, and this cofactor is going to join these folded polypeptide chains, and you will have now a functional protein. So what's the takeaway lesson? Well, the takeaway lesson is that the proteins are highly folded structures, and the folded structures is making it compact. Okay, not only compact, but you need to understand that this correct folding, making it compact, gives it a special, a particular shape. This is going to be important, and we'll study why later on, especially when we are talking about enzymes. So shape, compactness, correct folding go hand in hand for a protein. Please keep this in mind. Here I have given an example of hemoglobin. You'll study this in higher classes. In hemoglobin, there are 4 polypeptides, and these 4 polypeptides are joined together, and you end up, what happens is, with a functional hemoglobin, functional protein. All right, I think, yes, this is it. And if there are any questions or concerns, please call me. Thank you for giving me your time and attention, and I look forward to hearing from you. Thank you.